student.
All right. Well, I'd like to introduce Johnny Ubi. He's started working with me last fall, working on looking at a non-thesis project, a master's project. And I was working with some frog embryos with some snail eggs, and he got kind of interested in that. So I'm going to turn this over to him and let him explain what he worked on. This preliminary work from last semester that he's working on.
Sir, thank you. Hello, everyone. My name is Johnny Ubi Jr. Like you said, I'm a grad student in the biology department, and thank you for being here to listen to my research. So what I looked at was the toxic and teratogenic effects of the Pomacea maculata on the embryos of Xenopus laevis. So one, you might ask yourself, why these two organisms? And two, what are these two organisms? So the Pomacea maculata is an island apple snail that in recent years has inhabited the southern region of the United States and also parts of South America. The problem with these snails is that they cause pretty much a disruptive effect to any other mollusk in the area, and they become highly invasive. They're destroying crops. They're also fouling freshwater areas. And so another problem with these things is that they are highly reproductive and produce a lot of eggs, thousands upon every sitting. And so the things with these eggs is that they are bright pink. When they lay them, they're on docks, they're on walk walls, they're on rods, all sorts of places, and they're an eyesore. So one way to get rid of these is that they scrape them into the water. Problem, these eggs have been proven to be toxic in their egg and their egg casing. And so what we want to look at is if this toxic egg casing that's being distributed into these waters is causing any effect on the wildlife inhabiting these areas. Hence, we use the Xenopus laevis, which is an African clawed frog. They're perfect for experimentation because one, they live a long time, they're low maintenance, they can breed any time of the year, and also they have a similar genetic to humans, which is helpful in a lot of researching, especially dealing with both species. So, to set up my experiment, what we want to look at was to determine the lethal concentration of the snail egg toxin to developing Xenopus embryos. We also wanted to look at if it's causing malformations from exposure to the snail egg toxin. And we also wanted to determine if there was any effect on the growth rate of these developing Xenopus laevis embryos. So, as we set up our experiment, the first thing we did was took these frogs, breeded them so that they can give us eggs that we can use to test these things. So we set up the test, the eggs were collected, we uncoated them, and we separated them into different petri dishes that we concentrated with different levels of toxin. I used a control group and also used 6 different concentrations, starting at 0.01, 0.05, 0.1, 0.5, 1, and 2 of the concentration at milligrams per liter. So, as we went to decipher these eggs, we took healthy eggs. We decided each petri group would be 20 eggs per dish. We used 4 control dishes, totaling 80, and we used 2 dishes per concentration group at 40 eggs per concentration. So, in total, I ran this experiment twice. Each run is going to total you about 220 eggs approximately. So, as we created our protein extraction, what we did was we took these eggs, we used a process using a Downs homogenizer, which is going to uncoat these eggs and these proteins and grind them up. We took them through a centrifuger, and what that did is we ran it at 3,000 RPM for 30 minutes, and all this did was give us a supernate of the concentrated toxin that we can use for these experiments. So, the first results that I have here is I looked at the mortality versus malformation and got percentages for those. And so you can see my graphs here for my test 1 and my test 2. From my control group all the way to my concentration group of about 0.5, the percentages range from about 0 to high as 10%. It actually stayed pretty constant at a low rate. Starting at about concentration of 0.5 and a 1 concentration at my second test, I see a spike in mortality, but nothing for real with the malformation. So that tells us, okay, we're seeing a very toxic level, but as for malformation, not so much. That percentage stayed at a low level. The next testing that I looked at was the growth rate. I have test 1 here and test 2 with my bar graphs. And so we see a height of about 10.6 millimeters down to a range of approximately 9.4 in my first test, and about 10.7 to a range of about 9.9. So nothing major, but when we cipher into it, we can see that there is a trend and a decrease in its growth rate from our low concentration to our high concentration. I also looked at the pH and kept control of that with the environment just to see what was going on. We see a dip between our highest concentration and also with our blue bar, which is our control group. We measured that pH and saw that that stayed fairly consistent also. And that just kind of told us the climate of our dishes that we're dealing with. So at the end, so as far as data analysis, we tested and registered LC50, which is going to be our lethal concentration at 50% of our population at 0.68. And so that's saying that at a concentration of 0.68, it's going to kill 50% of our population. So we can definitely say that this toxin is toxic to these environmental species, specifically the Xenopus laevis. Our EC50, which is looking at our malformation at 50% of the population, registered at 9.5. That's a very high concentration for a test that didn't even run that high. So we can definitely say that there is no malformation effect that we can see so far that this snail egg toxin is going to cause on our wildlife, or per se, the Xenopus laevis. And TI, which is our teratogenic index, which is when you divide the LC50 by the EC50, registered at 0.07. Anything registered lower than 1.0 is considered non-teratogenic. So with a score of 0.07, we can say that this has no teratogenic effect so far whatsoever. And I have some pictures here just showing some of the malformations and what we saw when we tested. Here I have a normal looking Xenopus laevis after its 96-day trial. Here you see bleeding behind the gut area showing hemorrhaging as a malformation. This picture here, we see a stunted growth in the body. This one here, we see edema, swelling of the head, and we see some gut malformations. Our top picture here, we see the gut completely developing external to the body cavity outside of the actual tap hole. And that's my presentation.
Questions for Johnny?
I'll throw one in.
Okay.
Sure. So your LC50 was what?
So I had LC50 of 0.68
0.68 what?
milligrams per liter.
Milligrams per liter of the snail protein, right?
Yes, sir. Snail protein concentration.
Mostly just wanted to clarify. Okay, so you said it was highly toxic, but not, it doesn't not malform?
Yes.
Okay, I was wondering if that was because it was so toxic, maybe they didn't have a chance to malform because that's going. Or if that was,
So that's something that we could say maybe in the higher concentrations in which it killed them almost immediately, but for a lot of the lower concentrations, even though it didn't kill it throughout the whole 96-hour process, we didn't see any malformations forming. So it's safe to say.
so you said in the beginning the eggs were dangerous. How is the eggs are dangerous?
Okay, so we know through literature that's already published that these eggs contain a toxin, then either the egg or the egg casing. So we wanted to test that and see how toxic it actually was, at what levels, and if it showed actual teratogenic effects.
I don't know if you've noticed, but whether their behavior any different. So they might not have been malformed at lower levels of toxicity, but I didn't know if they were behaving differently or not. It might not have been something you even look at, but.
What happens when behavioral analysis come to your?
I got you. Maybe anything after 96 hours, or if we actually viewed them outside of a petri dish for that, I'd have an answer for you, but as far as I know, not so much.
That's not something normal people look at.
Okay, I'll let you handle that one.
Let's thank our speaker again. Is Andrew here?
Yes, ma'am.
Okay. I've got clips, it will slide down. Yeah, slide this one over so that this thing is on the edge, and then we'll clip it. Just as long as it was in. And then maybe move this one over a little bit.